Okay. Yeah, so today's lecture will be like kind of shortish, and so the so what I did was I kind of just going through over some of the textbook materials that I thought might be important, and uh, since it's a undergrad course, you don't have to go through all the details as in the textbook. So just I would say that just go over the main kind of important concepts and what's going on. And then next week, what we'll do is so in this class. So last week I said that the cell membranes of neurons are quite important, and that decides how the action potential or the actual message that goes through neurons and that gives us a behavior. That's where it's generated to the action potential, and it's primarily dependent on the properties of the membrane. So we'll just talk about that a bit. And in the next class, so Tuesday, Wednesday, Thursday, we'll go through one chapter each, and I'll also show some experiments that we can do in the laboratory. Not necessarily done in my laboratory, but that's are done by scientists all over the world. For today, just as I said, we'll go to the kind of the conceptual areas kind of shortly. The prototypical neuron we talked about, that you have a cell body, you have the dendrites where the information is coming in, and then the information is sent through the axon to the terminals and through the postsynaptic neurons. And we also talked about that the electrical signal goes down the axon, but there is a chemical event that happens, which we'll talk about in the next class, uh, the synaptic transmission. So how the electrical becomes chemical and the chemicals becomes electrical. And we'll just go over what the cells, what are the basic mechanisms are, and then next week we'll talk about and focus on synapses. Uh, so why is it even important to understand all of this? As you can imagine, a very simple reflex action, which is basically the example given here, that you are walking barefoot on, say, on a floor, and there's a pin there, and you don't know the pin is lying there, and you step on it. And it hurts, and then you immediately withdraw your leg. And so that event that happened, that the moment it pricked your skin to the entire event that happened, the entire behavior that you displayed, kind of moving your leg immediately or running or kind of jumping away from the spot, took just a fraction of a second. So, and if you can think about it, it involves the neuron that's innervating the skin of the feet, and the cell body is here near the spinal cord. So it had to go that far, then it had to talk to a motor neuron that would actually make you withdraw. So, and it had to have, all that had to happen in fraction of a second. So if you understand that, we'll generally understand the basics of neurotransmission. And that's kind of our goal here. So as we talked about also in the last class, that neurons are full of solutions. So there's a lot of all around water in the nervous system, outside the cell and inside the cells, and these are full of charged ions. And so since there's a lot of water, the ionic properties of water matter, and the fact that the water is the polar solvent, and things like sodium chloride, and there's a lot of salt, sodium chloride, potassium chloride, calcium chloride, and so a lot of salt in the cellular solution or extracellular solutions, and they need to dissolve in water. And since the polar solvent, the water dissolves most of it, and from the charged molecules, which actually becomes the kind of fundamental component of neurotransmission, because these are determining the potentials, that is the voltage difference between across the membranes. And one of the most important component that decides all of this is the cell membrane of the neuron. So if you know about the hydrophobic molecules and the hydrophilic philic molecules, you would know that the cell membrane, if you might have gone through in cell biology, that the cell membrane constitutes of this hydrophilic outside and hydrophobic inside. So it's almost nothing dissolved in water can pass through this. But then later we'll understand or try to go through that, there are molecules that are embedded, or the proteins that are embedded in this membrane, which selectively allow certain molecules to go in. And that's how the potential difference is maintained, the electrical signal is transmitted, and also it also gives obviously a structural stability to the cell. And as I was saying, that there are channels and pumps. So these are basically proteins that are, as we were talking about last class, there are transcribes they're coded by the genes in the nucleus, the genetic code in the nucleus, and it's transcribed and it's translated. And through the rough ER, it goes through rough ER, and then it goes through the smooth ER and the Golgi body, and it's processed, it's folded, and then eventually it's inserted in this into the lipid bilayer. So as such, nothing passes, but depending on the kind of protein that's on the bilayer, selectively ions can go in both directions. And so how is decided what kind of proteins they will allow? It depends on they form these things called ion channels. So the ion channels are proteins which are sitting across the membrane and allowing the ions to go either this way or that way. They have subunits, so like they are structurally very complex, and each channel can be comprised of several subunits, which are encoded by gene by itself, and they're assembled there. Depending on the amino acid sequences, they will decide what kind of ions or what. It's a part of potassium, or it's calcium, or it's chloride. It will depend on the proteins that are making this channel. So there's some construction going on next to me. Can you hear me? Hello?
Yes, we can hear you. We can hear you. Though sometimes it has become very loud.
One second. Yeah, can you hear me?
Yes, sir.
Okay. So there is some construction work going on outside the window. Anyway, so yeah. And then how would so as I said, there are channels, and then how would the exchange of ions happen between the membrane from both sides, inside and outside? So one of the ways it can happen is just by diffusion, which is concentration and temperature dependent, and the ions move through the channels depending on which side the concentration is more, and it's basically looking for the equilibrium. But as I said, that the neurotransmitter is very fast, and if it has to naturally, like by diffusion, it's much a slower process. So you need something much faster, and that's where the kind of the electrical mode of transmission comes into play, where there is a potential difference within the two membranes. And these channels now selectively allows what it would like to go through and when it's like to go through. So that's how the exchange happens, and then it's much faster, and it's also much more selective. And then so this is so the entire nervous system is the neurons needs to be, so if we placed our feet on the pin, and the neuron has to be very specifically at that moment get excited, and it needs to get silent, because otherwise we'll be continuously feeling sensing the pain of stepping on the pin. So we have to the neurons have to be silent otherwise, and only get activated when you stepped on the pin. Otherwise, if it goes on hurting, then the neuron goes on firing, it will go on hurting, and then it will be unbearable. So it has to be temporarily tuned to us stepping on that pin. So I mean, this system here is trying to warn us, okay, like there's a pin line there, so you should be very careful that you should be, like, if you're walking barefoot on that place, you should look for all the pins, and that's what it's trying to tell us. And for that, it has to hurt really bad, because if it doesn't hurt, then you're going to injure yourself. So it has to be under silent as such, but only excited when there is a stimuli, right? And the stimuli is very specific because there are millions of neurons. So the stimuli has to be very specific to the particular kind of neuron. So that's why it has to be silent. And so this is called the resting membrane potential, which is the inside. So if you basically recording you, so this technique called electrophysiology, where you take a glass pipette and fill up with potassium chloride, and you are poking a hole inside in the membrane, and you're recording from inside. So you're making the salt basically is very similar in nature to the inside the neuron. And then when you're recording from it and you have a reference like electrode, because it's a currently ground, so you can compare with that, and you will find that in the voltmeter that is around minus 65 millivolts. So that's what is known as the resting membrane potential. And so here, as I said, there is a resting potential inside is minus 65 volts. And so there is a inside cell, outside cell. Now there are potassiums or other ions here, and then there are channels. And what it's trying to do is kind of make it so it moves faster. And so the selective channels are deciding what is the ionic concentration on both sides of this membrane, or the cell membrane here. So as I said, so what happens in action potential, what you're going to see is that when the action potential comes, or when there's electrical transmission, you can have again an electrode filled with potassium chloride inside the cell, and you have a reference electrode that is ground. And what you're going to see the difference between both of them, and what you're going to see is something like this, where in the oscilloscope you're basically looking at the change in voltage, and you will see as the action potential comes and goes, there'll be an increase in voltage, and that will drop. So as the ones that you were recording from inside, the voltage is increasing, and it's decreasing and coming back if you're looking at the oscilloscope from connected with the outside electrode, the extracellular electrode here. So, and what would happen now? So that is when the action potential is generated naturally. So, there's action that, it's connected to another neuron, and the neuron is sending the information, and the action potential is generated here. What will happen if you artificially inject a current? So you're recording that inside voltage, but you are injecting current. What is going to happen? So if you see here, this is when the current is injected, and the voltage, this is the voltage changes. So the action potential basically one potential to, so the frequency is dependent on the amount of current that is injected, and that it's much better explained here in this figure, where as you increase the current, the frequency increases, but it has to kind of get to a threshold. So the amount of current is injected. So this is the threshold. So you will start getting action potential when you inject that amount of current. Yeah, and then if you would look at like very recent development. So as I said, like in next lectures, not the next week, but next week, I'll show you some experiments that we have done in the lab, and that we use this technique called optogenetic stimulation. So here you are artificially injecting current, where you actually you physically injecting current, and also this can be done with light, which is, so these channels are light-gated channels. So these are found in bacteria and archaeorhodopsins, and then in archaebacteria. And what they do is that you express them in the cells, and they are light-gated. So you can open the channels just by shining different wavelengths of light, and then the channels will open, and it will allow the ions to get through the membrane. And doing this, you can either activate a neuron or silence the neurons just by changing the ion concentration inside the cell. So, and why is this important? Because, so eventually, like our nervous system, what is geared for is generating behavior. So the action potential that I was talking about will eventually generate a specific behavior. And if you want to, if you give an electrical stimuli, you might be, injecting current in all the cells in the brain, but using very specific proteins, expressing the very specific proteins like that, you might be able to control where you deliver light in a very small group of cells. For the details for that, again, will come later. And then the next question is the action potential transmission. So the action potential is transmitted across the axon, and you can see that it goes from one direction to the other, and this is decided by the direction is decided by the change in potential across the membrane. Now, as I said, that it has to go much faster and it has to be regulated, and different kind of neurons need different kind of speed because different kind of information needs to be transmitted either faster or slower. So so that's why we have this myelination. So we are talking about oligodendrocytes last week, and the oligodendrocytes are basically creating these myelin sheaths around the axons. And so now, because it's a kind of insulation, so the action potential is not being able to directly go like this. So it has to kind of jump here. So the longer that is, this myelin sheath is, so these gaps are called nodes of Ranvier. So that basically it has to jump like this. So that is so that is basically deciding the speed of it. So the amount of myelination. So the more the myelination is, the faster the transmission is. So and again, we'll come this later. So like for an example. So so you stepped on a pin, and versus you stepped on, like, say, a pillow, which is very comfortable. The thing is, the pin information and the pillow information cannot go at the same time. So you're feeling both the sensations at the same time, but through the same area, but the sensation of the pillow, like something that's gentle and comforting to you, needs to come much faster than something that a pinprick would do. Because if it doesn't do that, when there is a graded sensation, which means that, say, you take a pin and I'm piercing your skin. So first, when I touch the pin on your skin, you just felt the tip of the pin. But as I'm slowly pushing into it, it's hurting. So, but when you're doing this graded kind of, you're purposefully pricking like that. So the information of the touch, that you've just taken the pin and touched the skin, that has to go faster. So that's why there are different kind of neurons, and it's innervating the same piece of skin, and it's basically telling the brain, okay, so first it's just touched and then slowly it's breaking. So that's why the neurons have different kind of myelination, so which information needs to go faster, and it's basically dependent on that. So with that, so as I said, so this will be very short, and I didn't get into much detail. So if I can show you the yeah, so I'll just basically, 1 minute, I'll show you the textbook that we're talking about. I sent it to you guys last week, day before yesterday. Yeah, I don't know how to change the screen now, but anyway, so what I would suggest is just so I'll send you the slides, and just whatever we talked about, just read a little bit of. You don't have to go through because this is written for more for a PhD program, neuroscience PhD. So you don't have to go through all the details of it, and just just read about the major concepts that we talked about. Such as the action potential and the nature of action, the properties of action potential. So these are pretty easy to understand, and the different phases of action potential and how the ions are changing, that is causing the transmission of action potential, and also the properties of the cell membranes, and what are the different channels and different kinds of channels and different kinds of pumps, and what are the difference between the channels and the pumps. And so that's pretty much it. So that will be very much easier version of it. And then we will move into synaptic transmission next week, which will get into much in like more details than today. And then we'll move on to development, which will again get into a little more details. So in synaptic transmitters, we also will try to keep it relatively simple. We won't go through all the details and just go, since it's and go through the major concepts. Okay, yeah. So that's it. And if you have any questions.
So how exactly does numbness work? I mean,
Oh, so anesthesia, like so basically you are silencing. So if you what do you call that? So it's basically silencing the neuron. So if you're silencing the channels there, so if you have, if you have any drug that basically closing these channels, then there won't be any action potential, even though you're trying to stimulate them. And so that's what it does, and you won't feel anything. Which would be something like a local anesthetic, right? So when you go to the dentist and they spray lidocaine, and then when you spray the lidocaine, it's basically closing the channels, and then you don't feel anything after that.
what flows in there when you sleep on your hand or something for a long time, your hand becomes numb. In that case, what is blocking the channels? What?
Well, that basically you're pinching, like physically, like you're pinching this thing itself. So not always it does, right? So sometimes if you like sit in a weird position, it can block your like physically block your nerve.
Okay, so something it's just like something like a contraction which blocks it off. No, nothing, no fluid flowing.
Right, right. Not fluid flowing, like you're kind of like temporarily not damaging it, but you're temporarily actually disturbing it. Like not how to say it. You're temporarily like physically like if you kind of physically, I mean, I wouldn't say like nothing goes through it, like you're not killing it or anything, right? But on the you're basically physically damaging it, kind of temporarily damaging it, which it can come back normally. Does that make sense?
Yes, sir.
I
Yeah.
haven't thought much, but I don't think you were blocking a specific kind of channels or anything. Yeah, anybody else?
Good morning, Professor.
Yeah.
So I was just asking whether, you said you'll be covering all these topics in future lectures, right?
Which topics?
The topics which you listed in this slide?
Yeah, so no, as I said, that we'll go through like the very basics of it, and then you can read about it. So we'll move into synaptic transmission. So we'll come back to some of this, not all of it.
Okay, sir. Okay. So you'll be covering these in greater detail in. So could you possibly send like a timeline of when you'll be covering what topics?
Yeah, so the next will be synaptic transmission.
Yeah.
So then the Tuesday would be synaptic transmission, and Wednesday would be neuro development, and Thursday and the next Monday would be basically sensation and perception.
Okay, sir. Thank you.
Yeah, so the development will take at least 1.5 class, and the sensation and perception at least take 2 classes. So
Okay, sir.
we'll get into more details in class. All I'm saying is that we're kind of touch because since it's a like introductory class, so we'll go through we'll just get into like the major concepts and not get into much details of it. Since I think there are totally 10 lectures, and uh, the the next is the sensation and perception chapter, the motor system, all this will take kind of two classes. So we cannot spend too much time on one, right? And those will be much more interesting because I think we can show experiments. We can talk about experiments and what are the experiments you can do with animals and in the laboratory. So yeah.
Okay, sir. Got it. Thank you.
Yeah, yeah, welcome.
So when you had mentioned about greater myelination, does that
Yes.
mean greater myelination across the length of the axon or the thickness of the myelination?
Yeah, the thickness of the myelination, and the so one is thickness of the myelination, and the other is the distance between the two node of ranviers, right?
Yeah.
So, so the distance, the longer it is, the more it has to jump, and the faster it goes. So both, either the thickness and the length.
Okay, and one more thing I mentioned about the frequency being proportional to the input current.
Yes.
So, like, I mean, can you just explain a little more about it?
Yeah. So what it means. One second. Is as simple as the action potential it's not so the change in voltage so it will spike once and then it will come back to normal and then it will spike once again. So that's how the voltage change happens, but the current that is injected is kept at constant, so the constant amount of current is injected over time. And the potential change, the action potential, as long as you're not killing the neurons, you're injecting so much current, and the neuron is still firing. The pattern would be like that. Like it will the action potential lasts for a very short time, but the change in potential, but it will rise and fall like that. But the frequency, like the gap between two considered action potential will depend on the amount of current that's been injected. Because that is the amount of current is changing the difference in potential, right? So the more rapidly it's changing the difference in potential, the more rapidly the action potentials happen.
Yeah, thank you.
So, in one of the slides, you talked about optogenic simulation and the fact that it was expressed like it was obtained from archaebacteria, right? Is there any neuron in our body or like in human beings or mammals which is also optogenetically stimulated?
Yes, so like mammalian, so humans is not done yet, but it can be done. And so mammalian cells can, so that was a challenge, right? So they were discovered from these bacteria. And then people thought it can, so the idea was that you artificially kind of remote control with light, you excite the neurons, right? And so the idea was that would be the perfect way to test what is the actual function of a neuron. So if you take one neuron, express optogenetic, so you can inject electric current in one neuron, but that's much harder in a living animal because the animal is moving. Where light is much easier because you can just shine light on it, and the specificities decided by how much protein or where and when the protein is expressed in that neuron. So that's why people wanted. The idea has been there for years, and they first tested in they were testing in C. elegans and worms and stuff, invertebrates. But because how these proteins are made, the machinery that requires to be this to be translated and folded properly, as we were talking about last class, that are known to be not known to be not they do not exist in mammals. That's what the idea was, and so people thought it will never work in mammals. It can be only done in invertebrates. But the more recent experiments have found that actually mammals can synthesize and fold it perfectly. So that fear was unnecessary, and then they found. So in mouse, we can show, as I said, we can show experiments. I'll show you guys experiments that actually we did. where you shine light and the mice feel pain instead of actually pricking the mice. So yeah, so and in humans it's not been done, but there are people who are trying to. It's more of an approval issue that are the regulatory bodies willing to approve this to be kind of injected or so you have to use genetic engineering to express these channels in human tissue, and so that is more of the question. So it can be done, but is the risk kind of worth it, the benefits of it? So that's where is that at. But in mammals, in monkeys, it can be done. In mice, rats, all kind of animals. Yeah.
And also a follow-up on that, was it surprising that, mammals were able to fold these proteins?
Yeah, it was surprising because the proteins that known to be like folding these proteins didn't exist in mammals, but mammals seem to have like kind of parallel machinery which they can fold it even without the specific folding machinery that are studied in other organisms. It is
Thank
surprising
you.
in a sense. Yeah, it is not solved by actually finding the mammals have the machinery or not. It was just found by just trying it. So they just tried it in mammals and it worked. And so they thought, oh, okay. So it's more of that than So people always thought it cannot happen, but it actually did, and people have tried it happened. Yeah. So anybody else?
Yeah, sir, I had a doubt.
Okay.
So, regarding Nikita's question, wouldn't the
Yeah.
photoreceptor cells in the retina, which does phototransduction, since it's of neurological origin, wouldn't that be a cell which is optically sensitive?
Oh yeah, so the rhodopsins are the eye. There's a GPCR. So like GPCR, so there's another thing. So they're different kind of proteins. So GPCRs are much slower and channels are much faster. So because you want to make it happen really fast, so you need a channel for that.
Okay. So, the second question is, in the slide where you were talking about artificially inducing an action potential, the electrode which is connected to the neuron, was that connected at the axon hillock?
The axon hillock. Yeah.
Okay.
No, so it
So,
was connected inside.
so could you repeat that? There was a slight
It was
disturbance.
connected inside the cell, right? Let me get to that. Yeah, so it was getting injected inside the cell, and the potential is also it is near the axon hillock, and then your potential is also being recorded from there.
Okay, sir. So, and a second thing is that, so in such uh, an experiment when we are artificially stimulating the neuron, we would be able to observe the refractory period of the neuron, right?
What was that? Sorry.
The refractory period between two action potentials.
Yeah.
So could we observe that in this experiment?
So I mean, at the cellular level, yes, right. But if you see the graph, it's almost immediate, right? So because the electrical transmission is really fast.
Yeah. So,
Yeah.
what kind of experiment would require us to measure that kind of values of?
I mean, this is the fastest we can record, right? Like, so the activity, this is the voltage changes. I actually don't know about it. I have to look into it, but I don't know the answer for that.
Okay. So, and this is just a request, but along with your lectures, could you possibly tell us about some research areas which are neuroscience? That
Absolutely. So the reason, so the for the next class, we'll see a lot of that. Uh,
okay.
yeah. So the major reason for that a lot of today's stuff about stuff that is covered in other cell biology classes, except for action potential and voltage kind of. But yeah, yeah, but the next class we'll see a lot of that.
Sir, could you repeat that because there was a lot of background noise?
Yeah, it's a lot of background
Yeah,
noise.
Professor, sorry to interrupt if you don't mind. Okay, yeah, the person has been muted. Sorry.
Yeah.
Yeah, I can't actually. Yeah. So anyway, so the yeah, for the next class you will basically see that a lot, right? That's what I'm saying. So next two classes, so next week all the class you will see that a lot. I'm not electrical physiologist, so like I didn't want you to show things that I don't really understand. So that is the reason I haven't done it.
Okay, sir. Thank you, sir.
Yeah,
Sir? Hello.
yeah. Tell me.
So, the optogenetic stimulation works in a manner similar to neurotransmitters, right? Where it induces the opening of certain ion channels.
Right.
So, can it be used as a potential mechanism to reactivate neurons that for any reason are not being activated by the respective neurotransmitters?
Yeah, yeah. So that's a very good point. So for basically kind of not regeneration, but the neurons has been dead for a while, and can you try to use that? And so one of the first experiments that actually they actually tried to do that. So in mice that are blind, can you use channels to kind of activate that? It didn't. It doesn't work really well because like it's kind of you're working on the assumption that that would solve the problem. This by then by electric just giving electric shocks should do it too. And for a lot of when you have spinal cord injury and stuff like that, so even practicing doctors do give low kind of inject low amounts of currents into the basically give mild shocks to be to allow the tissue to recover faster. So optogen can be very useful for that, but I don't think it's been successful yet, and it's mostly in laboratory studies. Yeah.
Okay, thank you, sir.
Yeah. Anybody else? I cannot hear anything actually. Can you all hear me?
So we can hear you, yeah.
Yes, we can hear you.
Yeah. So any more questions? Okay, so if you don't have any more questions, so today, so that's we'll yeah, so that we'll stop here today, and the next week it'll be more intense. So you guys can read a little more about the PDFs and about the action potentials, as I said, these experiments here, current injections, and also the ionic gradient, and about you don't have to read much about how proteins are. I think here they had how proteins, these amino acids are made and stuff like that, but just kind of superficially study about this, and also the myelination and the action potential transmission. That's at the very end. And so, okay, with that, I'll just stop it here. Okay, all right.
Thank you, sir.
Thanks.
All right.